Welcome students. This is the lecture number 2 for volatile oils, which we have discussed in the lecture number 1 regarding the definitions and various types of extraction, isolation, and how to isolate volatile oils from the crude drugs. And this is not only for the Pharm D students, even for 5th semester students, and even it will be helpful for the diploma in pharmacy students also to have a revision in their what they have studied. So you see here we are going to see some drugs, especially in the lecture number 2, and as I told you, I need 3 lectures for volatile oils according to the syllabus by Kuhas. Here today we are going to discuss some of the drugs which are very commonly repeatedly asked questions also we have discussed in the last class detailing for many university questions, which all the questions repeatedly asked also we have discussed in this Mentha, Clove, cinnamon, Fennel, Coriander, today's class. It's little wider. You can listen and you write yourself and make a notes, because some of them you have done in the lab, so it will be interesting for you to know little more points which I am giving, or little more points which you have. You can go club together, and if you have any doubt you can ask with me in my office. I can clarify you and I can give you more ideas or more references. I can support you by giving the points to be added. Now peppermint and peppermint oil, it is interesting plant which we are using even in our normal tea and coffee. When we are having, we used to take these leaves to dip in our coffee tea. So definition, when you want to say definition or a biological source, peppermint is a dried leaf of Mentha piperita, family Labiate. It should contain at least of 1.2% of volatile oil. Geographical source Europe, America, even in England, and we have in India also. And you can see macroscopical features. It is a beautiful flower, but we'll use to take the leaves actually. Flowers also very beautiful. It have the content of menthol. Leaves, you can see the dark colored leaves. Shade dried. The volatile oil may not be lost by when you are doing shade drying. All mints, that means mint, we used to say mint leaves. Mint have a square stem. When you see the stem, it likes a square stem, creeping rhizome. It has a creeping rhizome in the underground. And black mint, which is most common cultivated variety in England, has purple stem. You can see there is a 2 varieties I have taken in the picture to show you. One is green color and is purple color stem. The leaf blades are 3 to 9 centimeter long with a grooved petiolated or petiolate up to 1 centimeter long. Pinnate venation. Venation, when you see the leaf, you will have a pinnate venation. Granular trichomes. It contains granular trichomes. Bright yellow point by means of seeing by hand lens when you see, or an dissection microscope. And you see, you can see that very clearly, bright yellow points. And small purple flower appears in late summer. Just a simple description over. Microscopic features. They have that is diacytic stomata. Even you have the cellular structure question also they may ask. So you should have an idea about all the stomatas. The respiratory part of the plant is stomata in the leaf. So you can see anemocytic stomata is there, anisocytic stomata. You can see the multi-cells of 3 cells and 4 cells present. But diastatic, the diagonal towards the hole or the stomatous cell, you can see that shaded cell. So it is called diacytic. It is diagonal to the cells of the present. If it is parasitic like in senna leaf, you can see parasitic stomata. It will be parallel to the shaded cells of the stomata. And you can see parasitic, another form, and actinocytic, multi-cells surrounded the stomata, and you can see gramineous and tetracytic. You should know about these types of stomata. If suppose they are asking about the cellular constituents, different cellular structures of leaf, if they are asking any short notes, the students should be able to write these things. So that's why I have shown. But here in this Mentha piperita leaf, you can see that diacytic stomata. Multicellular clothing trichomes are present, in which there are 2 types of glandular trichomes which you have observed. One with an unicellular head like this, unicellular, only one cell as a head, and sometimes you can see multicellular head also as shown in the picture. So there are 2 types of trichomes present in the leaf structure. Calcium oxalate crystals is absent in Mentha piperita. And you can see this, the synonym, you can say Oleum menthae or mentha. It contains usually menthol, cineol structure. Other constituents also there. In the next slide we will have a discussion about that. But menthol, cineol, students can remember. That's why I am repeatedly added these structures of these compounds. It is the oil obtained by the distillation of Mentha piperita belonging to the family Labiate. Oil should contain at least 44% menthol, 15 to 32% of menthone, and 4.5 to 10% methyl acetate, and some quantity of isovarulate and cineol also present. And when we come to production of menthoil from Mentha piperita, the plants are dried in air. Sun drying causes the loss of active principle, that is volatile oil. The air dry in the shade, air dry material is kept in iron or steel vessels which has a false perforated bottom as shown in the picture. False perforated bottom in a cylindrical vessel or square vessel. You can have by air drying. The steam under pressure is passed through the drug for about 3 to 4 hours for complete distillation. Near about 80% of the oil is distilled off in the first half of the distillation itself. The medicinally important menthol distilled out in later half of distillation. The mentha oil is collected in separated vessel where it is decanted and filtered. The average yield of oil is approximately 0.5 to 1% volume by weight. This oleum mentha or Mentha piperita constituents, as we have discussed, menthol and jasmonone and menthone and pulegon and menthofuran. extra I have added. Menthol structure we have already seen. Jasmonone, slightly the same structure with a substituent. I don't want to waste my time. I will give the, you can get the jasmine structure also. Menthone, menthol, isomenthone, methyl alcohol acetate, lemonone is also there, lemonine, and cineol, menthofuran, pulegon, jasmonone are present. Jasmone is present. And the chemical test, take few ml of oil, mentha oil, mix it with 5 ml of nitric acid solution. What is this nitric acid solution? I have given in the bracket. The solution should be made in such a way, add 1 ml of nitric acid in 300 ml of glacial acetic acid. From that solution, you have to add 5 ml to the oil. Heat the solution on a water bath. For 5 to 10 minutes, the liquid develops blue color as shown in the picture. It gets deep blue color upon more heating and shows copper blued fluorescence as shown in the picture. And again, after few minutes, it turns to golden yellow color. This is the specific test for mentha oil. Uses: reduce spasm and pain caused by endoscopy. If somebody is undergoing an endoscopy experiment, that pain can be relieved by means of this. It can relieve migraine, headache, treated for nausea, to treat bad breath and reduce dental plague. It helps to heal ulcer. It relaxes muscle in gastrointestinal tract and relieve colon spasm. Sometimes people will have an abdominal spasm due to colon pain. Large intestine colon pain can be relieved by this menthol. Menthol kills bacteria and parasites. You can see the mechanism of action also when you want to know. Menthol acts as a calcium channel blocker. It affects the smooth muscles of vessels of GEOGRAPHICAL on neuron cells and heart. By means of that, it can kill bacteria. It blocks the calcium channel and prevents the influx of the calcium channel and leads to relaxation of the smooth muscles. So just I want to add this one as a mechanism of action, means we cannot add this. It affects the smooth muscle of GIT or the neuron cells of heart. We cannot say to the pharmacological action. Even it is telling that it acts as it kills the bacteria and parasites by means of acting in them in this way. That's why I have added this mechanism of action. In fact, I hope even this part even we can remove from our slide instead of making confusion for the students. So it can act as a killing as a bacteria can be said very clearly. That's what the uses. That is enough for you to write instead of making to in a wrong way answer. Doses you can see as infusion, not infection, infusion. Take 3 to 6 grams of peppermint leaves in a hot water and covered with for 10 minutes. You can keep it covered and use twice a day. And a capsule, take 0.6 ml of peppermint oil, split into 3 doses. You can split it in them, and 0.2 ml per day. For headache you can take 10% of peppermint oil in ethanol solution. Apply across the forehead, repeated for 15 to 30 minutes. Good relief you can see the expected. Clove, you can see the clove picture, which is very common in our area, our place in the southern part of Kerala and also Kanyakumari district. We are having clove. You can see this is in repeated questions also. They are asking about clove in detail. So I have added the cultivation also. You can just have an idea and you can write so that it is easy for you to remember. Definition, or you can say biological source or botanical source of clove. Clove are the dried flower actually, flower buds you can say, of Syzygium aromaticum, or you can say the previous name Eugenia caryophyllus. Family Myrtaceae, the bracket I have said, Myrtaceae is the family. That is, you should know that Syzygium aromaticum is also the botanical source, and the plant can be called Eugenia caryophyllus. The previous name is also correct, Eugenia caryophyllus. Eugenol is the main constituent, Eugenia caryophyllus. Geographical source, it is native from the Molucca Islands and traditionally cultivated in Tanzania, Zanzibar, Madagascar and Ireland. Highly medicinal plants in Africa, Madagascar, Indonesia, Sri Lanka and India, the main Nilgiris hills, we have more cultivation, and Kanyakumari district, Kottayam, Koilon hills of Kerala, we have more cultivation for this clove. And we are very much useful for our various purpose, normal daily use. Cultivation, the deep, rich, loamy soil is good for clove cultivation, but it can also grow in sandy, loamy and laterite soil. It favors warm, humid climate with annual rainfall in the range of 150 to 250 centimeter. It grows well as at an altitude of 90 meter from the sea level. The seeds are sown on August to October for cultivation purpose. The seeds are sown in nursery bed at a distance of 10 centimeter. The seeds germinate in 4 or 5 weeks, and these seeds are transplanted to pots for 6 months. These seedlings are kept in pot for nearly about 12 months. 12 months it has to be kept in the pot. Then they are transferred into open field. The shade is provided. Shade should be provided. Why? It is initial stage because the plant cannot bear the full sunshine. It can also be cultivated along with other plants like arecanut, coconut and nutmeg. These all are some of the plants in between you can cultivate, you can plant this clove. The suitable fertilizers which are used like ammonium sulfate, sulfur phosphate, potash, etc. are provided to the plants in 2 doses. The first dose should be given in May or June and second dose in October. The plant started bearing after 7 to 8 years and gives abundant yield per acre after 15 to 20 years of growth. Generally, the clove plant produces about 3 to 4 kg of drug per year. The clove buds are hand picked. You can pick it by climbing with a ladder or beating with the bamboo. Sometimes platform ladders are used to go near to the flower buds to pluck them, used in the case of tall trees. And collection starts when the clove buds changes its color from green. This is the green color. Slightly changes to slight pink. You can start the collecting. The clove on sun dry or sun dried and kept free from foreign matter, it will turn like a crimson red color as shown in the picture, and then brown blackish color, brownish, dark brownish black color. That is the original clove bud. You can see this clove bud, how it is the color of the clove bud. Macroscopic character, color crimson red to brown. Characteristic or aromatic odor, taste aromatic and a pungent taste. Length 10 to 18 millimeter, width is 3 to 4 millimeter, and the thickness is 2 millimeter. Shape, flower buds are nail shaped. Hypandium, quadrangular length 10 to 12 millimeter, diameter to 2 to 3 millimeter. Head globrous head, you can see that. And hypandium corresponds to the inferior ovary. When you take the section, you can see very clearly. And an globule head, polished head, surrounded with 4 diverted sepals. You can see, even the picture can see in the sepal lobe, consists of 4 imbricated petals wrapped numerous incurved stamens will be present. You can see these stamens, anthers in the powder microscopy also. The pulverized powder, that means the powdered drug of the clove. The presence of large secretory cavities, calcium oxalate crystals, pollen grains, fiber with thick cells can be seen, lignified, pungent wall can also be seen in the powder microscopy. That is called pulverized. This is the TS. You have to draw very neat diagram when they ask essay question, 5 mark or 10 mark also. With pencil, don't draw pharmacognosy diagram with pen anytime. It's a mistake you are doing. No teacher will give you mark for you when you draw by pen. You have to draw the diagram with pencil. Floral diagrams should be drawn by pencil. So you see the whole structure in a 5x or 3x. That is the first diagram. Second one is enlarged diagram in the 10x we can see, or even 45x also we can see a part of the transverse section below the hypandium. You can see this clear structure of the cuticle, epidermis, oil cells, see the picture, and saprophyte. That is, saprophyte means calcium oxalate crystals can be seen in cells below that oil cells, oil glands. The layer of cells, parenchymatous cells or the polygonal cells inside, you can see that saprophyte. And the vascular bundle, the phloem and xylem together, pink lignified cells can be seen, vascular bundle, and pericyclic fibers, pinkish pericyclic fibers spots can be seen in the transverse section as shown in the picture. Parenchyma cells, this is peculiar, specific identification of microscopical character of the clove. Parenchyma cells, like a chain, you can see that. interesting and it's observed in the lab, you can see in the practical. And then the middle part, you can see the colonchema cells and the brown spots which is not mentioned, browny matters also will be there in the center part. So this you can explain clearly of the parts which is shown in the enlarged one. In the small structure in the top which I shown the same one, the middle colonchema, aerenchyma, cortex, the complete area below the epidermis, we have shown the cortex area. You can see that area, oil cells, presence of epidermis. You can mention very clearly in the structure. So what are the things which we have just discussed, you have to remember, write down and make your notes. Heavy cuticulated epidermis, numerous oil cells, they are called as lysolysigenous oil cells which are lysigenous. So you should write like that. And calcium oxalate, cluster of crystals, prisms are present. And stomata is also epidermal and sepals can be seen in the powder microscopy. And starch grains in the fruit of mother cloves can be seen little more. But we are not going to take mother clove, we are going to take the flower bud. Mother clove will be having less volatile principles. Lignified sclerenchyma, del cells also may be present apart from the structure which is shown in the microscopical structure. This is just for your understanding. Again, I have shown the picture. Constituents, you can see 14 to 21% of volatile oil. Mainly eugenol, isoeugenol and cytosterol, stigmasterol, camphosterol. Tannins are present. That is triterpenic acids and esters. Glycosides, some are present. But main constituents is eugenol. They are volatile oil containing eugenol. The properties of volatile oils, that is clove oil, color. Clove oil is colorless to pale yellow. Specific gravity 1.038 to 1.06. Refractive index cyanide by cyanar is 1.527 to 1.535. Boiling point 250 degrees centigrade. It should be stored in proper condition because it becomes dark and thick on storage. Chemical test, needle-shaped crystals can be seen in the microscope observation or even in the lens. Section taken of a clove and treated with, if you treat with strong potassium hydroxide solution. Very simple experiment. You can see needle-shaped crystals. Uses of clove. Clove-based phytomedicines are used locally to treat minor wounds after cleansing. As an analgesic, headache, toothache, you can keep a clove in the toothache as an analgesic in disease of mouth, pharynx or both, in form of lozenges, in mouthwashes you can see, and in oral hygiene. Internally it is used as a treatment of gastrointestinal disturbance like epigastric bloating, improper digestion and flatulence. Gripping problems can be removed. In Germany, clove-based products are used in mouthwashes for swelling in mouth and throat. In Indonesia, it is consumed as cigarette, that means kritik, it is called kritik. Cigarettes are available in Indonesia. Adulterants for clove. Number one, exhausted clove, the volatile oil is removed. That means oil free, they have taken oil and they will sell. That is adulteration. It doesn't have any quality without oil. Oil free or contain very less amount of oil, dark in color, shrinked and floats on the water. That is the adulterated clove. Clove stalk sometimes added for in the total volume to increase the weight. Generally used for adulterate of powdered clove. Ash value is high. If you see, check. Fiber content is high and calcium oxalate presence or present containing 5% of oil. And brown cloves also added, expanded flowers of clove tree and stamen free containing less volatile oil. This is also another mother clove. Late collection. Dark brown in color they will be and oval in shape. Slight aromatic, starchy, containing less volatile oil. Cinnamon, the next drug. Cinnamomum xylanicum. You might have done the bark experiment. Cinnamon and the carafa, or you can say that is which we are using very common in our non-veg preparations and other preparations. And it is, and also it is commonly in our place, you can see this plant available in our gardens. Beautiful stem with design. And you can see that cinnamon is a dark, or you can say it is dried bark or inner bark in Cinnamomum xylanicum laraceae. Geographical source, they are cultivated in Sri Lanka, South India and Seychelles in Madagascar in African island and Martinique and Cayenne and Jamaica and Brazil. Cultivation and collection and preparation. See that stem, how they have taken the outer bark and the inner bark will be dried. I have separate class for cinnamon, clove. You can just see that separate the drug classes in my YouTube. That will be with videos, how they are cultivating, collecting, drying, all these things can be seen. Interesting for you to know, and you can write your own notes and it will remember you. Here, according to the syllabus as a theoretical, I am just explaining for you to prepare a notes. Grow by seeds, cut down when 2 to 3 years old. After 5 to 6 shoot grows from the stump of 18 months, shoots are harvested, trimmed and fermented. The bark is removed, peeled bark is then stretched over a suitable stick. Out cortex is removed because it is an inner bark only we are using. This is the plant which is cultivated, and this is the time for them to remove the bark. This is the small plant and which the seedlings, and just to show you the height of the plant. Preparation of cinnamon is an interesting process. Quilled bark as shown in the picture. Individually scraped bark are placed inside each other. Compound curved, squilled, are dried on wooden frames in open air without exposed to direct sunlight. They will be hanged. You can see the video in my private or in separate cinnamon class without exposing directly to the sunlight, and sorted into grades also. Macroscopical features, there are broken which is available in the shops which we are using. Quilled one is the original one. Normally receive in shorter length known as cigar length. Contains single or double compound quill about 6 to 10 millimeter diameter and vary in length. Thickness of the quill varies according to grade. Good quality, not more than 0.5 millimeter thickness. Bark 10 to 40 millimeter. External surface is yellow brown, shining, wavy lines, pericyclic fibers, which we can see the outer area with, and occasionally scars and holes leave twin position. Inner surface is darker, longitudinal, striated. Odor fragrant. Taste warm, sweet, aromatic. This is the structure which you have to draw very clearly, stating the cork cells. Usually the cork may not be available, may not be present because it is an inner bark. It will be broken as shown in the figure. Inner bark means cortex area where starch grains or aileron grains which is present. Pericyclic fibers, scleroids fibers, pericyclic fibers pink in color, they are lignified. And phloem fibers, next part after scleroids cells which is pink in color, darker cells you can say, that is scleroids, dead cells, and phloem fibers and secondary phloem and medullary rays. You can see the medullary rays arrangement of the cells like a brick. Two medullary rays you can draw, and oil cells, and you can say even raphides can also be shown as shown in the picture. TS of cinnamon is very easy for anyone to draw. So microscopic transverse section, absence of epidermis and cork cells. Usually there is, as I told, absence, it will be broken. It's an inner bark. Cinchona there will be bark, cork cells will be there. It is outer bark, this is inner bark. So scleroids, thick lignified pitted walls. Secondary phloem is composed of phloem parenchyma containing oil, mucilage cells, phloem fibers, medullary rays. Some phloem parenchyma contain tannins, secretion cells, volatile oil and mucilage, calcium oxalate. So this is a structure. I am again showing, you should draw the diagram, medullary rays, oil cells and sclerenchymatic cells, and the cortex area. You should show the pericyclic fibers clearly as a lignified pink cells. Chemical constituents, especially cinchona bark, contains polycyclic diterpenes and cyanodianoid oligomers. It contains volatile oil of 0.5 to 1%, phlebotannins 1 to 2% or 1.2%. Mucilage is present, calcium oxalate, starch and mannitol responsible for the sweet taste of the cinnamon. Cinnamon oil obtained from distillation method, which is slightly yellow in color and upon storage changes into red in color. The essential oil 5 to 20 ml per kg is composed of phenyl propane derivatives, and this is a structure of cuminaldehyde, cinnamonaldehyde, which is present, very interesting and important structure, chemical structure. Eugenol is also present. Cinnamon oil mainly contain cinnamon aldehyde. You should write aldehyde. It's very small benzene with an aldehyde, cinnamon aldehyde, and 60 to 70%. Eugenol 5 to 10% only. Benzaldehyde, cuminaldehyde, and other terpenes such as flanandrine, pinene, cymerin, and caryophyllene. All these things you have to add, and clearly you should mention the chemical constituent. Chemical test: to a drop of volatile oil, add a drop of ferric chloride solution. A pale green color develops. Due to cinnamon aldehyde produces brown color, and Eugenol gives blue color, which results in the formation of pale green color. So you should know that why it is pale green color, because cinnamon aldehyde is also present and eugenol is also present in a low content. Cinnamon aldehyde will give brown color, eugenol is giving a blue color. Brown and blue, then finally it gives a pale green color. Uses: the drug is used as an aromatic stimulant, antibacterial, antifungal, antiseptic, carminative, and stomachic, and astringent. Commercially, it is used as a spice, condiment, candy preparation, and dentifriers and perfumery industry. Cinnamon oil is used in urinary tract infection and food technology. Cinnamon oil can also cause some allergic reaction also, cinnamon oil. Cinnamon oil and cinnamon aldehyde are irritant to some of them skin and mucosa membrane. They cause sometimes allergic reaction like urticaria or edema in the faces and lips for some persons. Cinnamon oil adulteration and allied herbs also you should know. Adulteration: cinnamon leaf oil can be mixed and oil of cassia can be added, and sometimes allied herbs like cayenne cinnamon or cinnamon xylanicum grown in Brazil, not used in Britain. Cinnamon laureri from Vietnam closely resembles cassia, Cinnamomum cassia. So that is also an allied drug which can be used. Fennel and coriander are umbelliferaceae fruits. You might have done in a chemical, in a practical also. Very, very important. Very, very important. Theoretically and practically also, students should study in detail and prepare a very good notes. Fennel, we know that we are commonly using this drug in our food. Fennel, umbelliferaceae fruits. You should know the parts. It looks like a seed. Everybody says seed, but it's not a seed, it's a fruit. Umbelliferaceae fruits. Fennel consists of dried ripe fruits of Forniculum vulgare, family Umbelliferaceae. Geographical sources: Europe, India, China, Egypt, Mediterranean origin. Fennel is indigenous to Mediterranean countries and Asia. It is largely cultivated in France, Saxony, Japan, Galicia, Russia, India, and Persia. This is the plant. Beautiful when it gives descendants of the fruit. Yellowish fruits can be seen like this. When it is ripened, you can see the area will be completely yellowish. Cultivation and collection: fennel is hardy, beautiful plant, perennial, umbelliferaceae herb with yellow flower and feathery leaves. Grows wild in many parts of the world. In many parts of the French people, they are taking the underground. The stem part is most delicious and used for salad, and that part is used. And also fruits is used, umbelliferaceae fruits. Stem part also most of the place, even I have shown in the separate fennel classes there in that video is added. You can see that how it is used, the whole plant having more importance in European countries. Fennel is propagated by seeds while cultivation during April in ordinary soil. How she is sowing the seed, you can see. The fennel requires abundant sunlight, and it adapted to dry in sunny situation. It does not call for heavily manured ground, but it will yield more on well-drained calcareous soil when you are using. You can see it is having about 4.5 or 4.5 to 5 pound or seeds or kilograms, little less of seeds are sown per acre, either drilling or by 15 inches apart. You can keep the seedlings as shown in the picture, even covered with soil. The plants grows to a height of 2 meters, erect and cylindrical, and take enough space in branching. And most of the branch bear leaf cuts into very finest of segment. Here in this type of plant which I shown, that white color in the picture which is shown, that's what I was telling, edible part, which is very commonly used for salad, and it is being used in European countries. In the same plant which we are talking about, the volatile oil, but that is the edible part is also very common and export quality. The plant bears fruit in the second year and the bright golden flowers as shown in the picture. Flat terminal umbel blooms in July and August. The fruit is collected by cutting the stems in September when the fruits are ripened. The stems are dried on sheaves under sun and later beaten to separate the fruits. They are beaten also, like paddy, how you are removing. Fennel fruits, you should know the picture very clearly. This is a part, you can see it is called a mericarp. When it is together, it is called cremocarp. The topmost part is stylopod, and the inner lines you can see the primary ridges, secondary ridges down like this, primary ridges, and pedicle. You can see the fruit is an entire cremocarp with a pedicle, oval, oblong, 5 to 10 millimeter long, 2 to 4 millimeter broad. It has greenish brown and yellowish brown color with 5 prominent primary ridges. Lines you can see when you remove 5, and a bifid stylophode like a cap on the apex part, which is shown in the picture. You see this picture? It is just drawn by separating the two mericarp. Cremocarp together, the red line in the top. Stylopod is part made into two. In between there is a joining that is called carbophore. When you take a section, you can see this carbophore as a round part in the middle, joining two mericarps. And this part is dorsal, and this is part is called commensal. We have seen the bones, you might have studied in anatomy, dorsal, ventral. Here instead of ventral you are saying commensal surface, inside surface. Outer is called dorsal surface. Very interesting part. You can draw the diagram. So two mericarps together they are called as cremocarp. When it is open, this part is called the dorsal area, this is called the commensal area. In between they are joined by one pedicle which is carbophore in the part. So when you take the section in the middle, you can see the carbophore. The lines which is shown, which is not marked there, they are called primary ridges and secondary ridges. 5 primary ridges are there in the dorsal surface. That's what the student can remember now very clearly. So when you see the TS, the picture you can see one part. This is only one part. That means it is the one part of mericarp. When it's 2 is there, means they are called as cremocarp. When you take a section, you might have seen in the lab, you will have 2 together in between. The carbophore will be there. Here only one carbophore in the bottom, and you can see the primary ridge like a hill, and down, that is secondary ridge, primary ridge. Each and every primary ridges you can see there is down, some cells are there, vascular bundles. Every secondary ridges you can see one vitae around, that is oil cells. Vitae is present in the secondary ridges. primary ridges, you can see there is vascular bundle and parquetry layers, and also pitted parenchyma will be there very clearly. And that area is called mesocarp. You know that when you see in a mango, you can see epicarp, mesocarp, endocarp, and there is the cartilagens. You will be separately, the seed will be there. So that is separate a fruit. But here umbelliferaceae fruit and seed are together. So you can see the outermost epidermis and the mesocarp, the fleshy area where you can see that primary ridge and secondary ridges, where the vitae are present in the secondary ridge, and the vascular bundle, parquetry cells, or you can see the pitted parenchyma, reticulate parenchyma, a special character of the fennel is present in the primary ridges as shown in the picture. You can see that. And then you can see the reticulate parenchyma also I have marked there. And you can see that next comes one line, that is a seed coat, that is called endocarp or testa area. The testa is the outermost hard part. The marking is not right. You can see the line, the testa, and the endocarp is inside. Testa is the line. It should be, the line is the testa. Then comes a raphe, a small raphe, and then the carbophore, and even the area there is two vitae are there. You can see two vitae. Can you see the picture? Very interesting. The student should see this picture while studying. You should know this is a mericarp part of the fennel. Cremocarp means two mericarp together is called cremocarp. Only one of this one is called mericarp. That section is shown here. So you can see that carbophore in the which surface, that is called commissural surface. And the other one is dorsal surface. In the dorsal surface you can see the epidermis. In the commissural surface, in the dorsal surface, how many vitae? 4 vitae are there. You can see there are 4 secondary ridges. All secondary ridges you have 4 vitae. In the commissural surface again 2 vitae. So there are 6 vitae in one mericarp of a fennel. In the whole fennel, so that you will get 12 vitae will be there in whole fennel. Because this is a half part which is the mericarp. This is cremocarp. So in one fruit of a fennel, if someone asks you question, how many vitae are there in one fruit of fennel? You have to tell that 12 vitae are there. And very clearly you can explain. 4 vitae are in the dorsal area and 2 vitae are in commissural surface of one mericarp. So there are 2 mericarps to form one cremocarp of fennel. So that 12 vitae are there. Every secondary ridges are having one vitae. And there are parquetry layers. Parquetry layer is special character for not for this, is also present, but special character parquetry layer is present for coriander. Here reticulate parenchyma, vascular bundles are present in the primary ridges, and they are very promptly pink in color, and pitted parenchymas are also seen. And you can see very clear carbophore and a testa and an endosperm. Aileron grains can be seen in aileron and in the endosperm area. So very clearly you can explain in detail the transverse section of mericarp region of a fennel shows 2 prompt surface of dorsal and a commissural surface. That's what I was told. Dorsal surface and a commissural surface. The commissural surface has the carbophore as shown in the picture, and 2 vitae are there. And the dorsal surface have total 5 ridges, 5 primary ridges you can write, 4 secondary ridges. The mericarp is divided into 2 pericarps consisting of epicarp and mesocarp, and testa and endocarp. Don't confuse with this word. This is about the fruit which you are talking about. This is not about mericarp and cremocarp. Mericarp, cremocarp is totally different. This is mericarp and this is cremocarp and there is two mericarp. This is about, we are talking about the epicarp, endocarp, mesocarp, about the fruit part. You can just even forget about that part. Especially epicarp consists of polygonal cells, this outermost. We are telling about the outermost. Epicarp means outermost part. Polygonal cells of epidermis, or you can say epidermis, which is tangential, elongated and covered by the cuticle. And the structure of reticulate parenchyma of the mesocarp area, where the ridge area, very good pitted parenchyma can be seen, pink color. That is mesocarp of the parenchyma cells of 5 bicollateral vascular bundles belong below each primary ridges. As I told, each primary ridges, a lignified, rectified, reticulate parenchyma sounds the vascular bundles which is present. It is pink in color and it is lignified cells. There are 4 vitae in the dorsal surface and 2 vitae in commissural surface, or you can say it is a ventral surface. And in epidermis or the endocarp shows parquetry arrangements, also seen a group of 4 or 5 cells in the powder microscopy which can be cleanly seen parallel or acute angles. Cells or similar cells, different direction can be seen. Even it is the special character can be seen in coriander. Even it can be seen even in the fennel also. Testa is the hardest part of the seed. It is umbelliferaceae fruit, so seed is also present inside the fruit along with the mesocarp area. So testa is a single layer tangentially elongated cells with yellowish color. Endosperm consists of thick-walled, wide polygonal or polyhedral colorless cells. Cells contain fixed oil, aileron grains and rosette crystals of calcium oxalate. So aileron grains is a starch grains which is present in the endosperm. It is a special vacuole where storage of protein accumulate in a stable form in the seed. They may ask, what is an aileron grain? It is a starch grain. It is a storage in the seeds. Usually it's an endosperm of the seed cells are containing aileron grains from the aileron layer. So this is a structure of parquetry layer cells. You can see an acute cells of parquetry layer which is seen in the 45x diagram, segments of endocarp which shows the parquetry of parenchyma presence in acute angle cells in opposite directions. This is a simple diagram to show about the pericarp and of one part of the mericarp and also the cells of TS of fruit of mericarp, which you can see as a picture. And this is a simple schematic diagram to show the reticulate parenchyma present in the ridges, primary ridge, vascular bundle present in the primary ridge, about raphe, carbophore, vitae, where is present. This can give you an idea for how the 6 vitae present in half part of a fennel. It's a structure of the fennel. Again, I have shown the picture. And this is the powder microscopy. When you see the powder microscopy of fennel, segments of epidermis cells of epicarp with a smooth cuticle and a well few anemocytic stomatas are also seen, and a fragment of lignified reticulate parenchyma of the mesocarp present in the section can be seen in the powder. Parquetry layer of the mesocarp can also be seen. Fragment of yellowish brown vitae can be seen, oil cells. And the fragment of thick-walled cells of polygonal endosperm cells containing globules of fixed oil and aileron grains, starch grains containing micro rosette crystals of calcium oxalate can also be seen in the powder microscopy of fennel when you are analyzing in 45x. Active principles or active constituents present in phenol are 4 to 5 volatile oil, trans anethol. Anethol is a constituent, and astragol, fancone, flavonoids, and coumarins, glycosides are also present in them. You can see fancone is an important constituent of phenol, and methicone as a methoxy group which is present in there. All of them are cyclic terpenes, monoterpenes. The best variety of phenol contains 4 to 5% of volatile oil. The primary constituents of volatile oil are 50 to 60% of anethol, phenolic esters, and 18 to 22% of fancone, a ketone. Fancone is chemically a bicyclic monoterpene, which is a colorless liquid, and a odor and taste is pungent and camphorous. The oil of phenol has beta-pinene, anisic acid, philandrin, and anisic aldehyde. Phenol also contains about 20% of fixed oil and 20% of proteins. Actions and uses of phenol is carbonative, expectorant, aromatic, all due to anethol and fancone. Uses in flatulence, excess gas in the stomach of the bowel can be expelled, and dyspepsia, indigestion, upset digestions can be solved. In chronic cough and cataract. Catarrh means is a buildup of mucus in the airways in the common cold activity of the body. It usually acts as a bag to the nose and the throat or a sinuses, and air-filled cavities in the bones of the face. It's often temporary, but some people experience it for months or years. This chronic problem can be solved by phenol. It is known as chronic catarrh. Phenol is used in stomachic, aromatic, diuretic, carminative, diaphoretic as a digestive, pectoral, and flavoring agent. Anethol may have estrogenic-like activity and habitate spasms in smooth muscles. Phenol has increasing production of bile, used in treatment of infant colic. Sometimes babies will go on, infant will crying, so the drops which contain phenol extract of anethol can solve this problem in infant. To promote menstruation in women, can increase lactation, acts as an antipyretic to reduce increased temperature, antimicrobial and anti-inflammatory action also is there. Adulterant phenol is generally adulterated with exhausted phenol, and due to its improper carrying during harvesting, they are also adulterated with sand, dirt, stem, weeds and seeds, etc. In which part of volatile oil is removed either by extraction with alcohol or steam distillation. Fruits exhausted by water or steam are dark in color, by that you can find out. Containing less essential oil and sink in water. They will sink in water, as we have seen in the clove how it floats, this will sink. But those exhausted by oil still holds 1 to 2% of oil in them. Coriander and coriander oil, today is the last drug which you want to discuss. Definition: coriander in the dried nearly ripe fruit of Coriander sativum. It's also umbelliferaceae fruits which you done in the lab as a section, so it will be easy for you to understand. Geographical sources: indigenous to Italy, also cultivated in Holland, Central and Eastern Europe, Mediterranean, Morocco, Malta and Egypt, China, India and Bangladesh. Macroscopical features, you know that it's like it's a little wider, same size, ball-like, stylopod. Primary ridges and secondary ridges are more than when compared to Fennel. As I told, the two part of mericarp together it is cremocarp, same. Annual herb going 0.7 millimeter in height with white and pink flower. Normally consists of whole cremocarp, strong yellow, 2 to 4 millimeter in diameter with ripe. Considerable variety occurs. Example, Indian variety is more oval. Apex has 2 styles. Fruits have an aromatic odor and spicy taste. Unripe plants, unpleasant, moissy odor. Same odor, oil, as when made from unripe fruit. This is the fruit, unripe. This is a ripe fruit picture of primary ridges, secondary ridges and mericarp and cremocarp. This is a TS which shows not very prompt primary ridges, but it has many primary ridges as shown in the picture. Vitaes are very less. You can see only 2 vitaes in their commissural surface. You should know what is commissural surface, what is the dorsal surface. In dorsal surface there is no vitaes. There are shown in the picture, it is not vitae, it is called lacunae. It's a vacuole, it is not having any oil cells. Vitaes or oil cells, only vitaes are present in, very less vitaes are present in coriander. You should understand, it is only 2 vitaes are present in the commissural surface or ventral surface as shown in the bottom, near to the carbophore, not like Fennel. That is the biggest difference. And you can see there is no primary ridges containing vascular bundle and reticulate parenchyma or pitted parenchymas are not there. Here you can see a chain like a line. You can see a line. What is it is, right? It is called tangential lignified fiber. Tangential lignified fiber, longitudinal lignified fiber, and it is showing a very interesting lines as a pink color chain, longitudinal or tangential lignified fiber, and you can see very clearly in between. You can see in that area only the vascular bundles will be present, and all these things, the chain can be seen. And aileron grains containing endosperm is same, testa is same, and only thing is vitae is very less in coriander. Only 2 vitae are present in one mericarp. Mericarp means one part. Even this is also like this, 2 parts will be there. In one mericarp, you will have only 2 vitae in the commissural surface near to the both sides of the carbophore. And raphe is same present, and endosperm, testa, and endocarp, that is about mesocarp, and endocarp are fruit part, and epicarp is the outermost part, that is a pericarp. But we are telling about mericarp and cremocarp is different. That is called mericarp means together that parts. One part is called mericarp. This whole is called mericarp. But there the parts is said is pericarp means epicarp, mesocarp, endocarp, that is the parts of the fruit is collectively called pericarp. That part is different and that other one is different. Just you can 1 or 2 times when you listen to this class, you can understand, and clearly you can write about the parquetry layer and about the longitudinal lignified fibers and tangential lignified fibers, how elongately we have seen that chain like there will be a longitudinal and also tangential pinkish color of a chain like parenchyma cells can be seen with a pinkish color. And this is a structure, once more I have shown for the students to know to draw clearly with a pencil if they ask in any 5 mark or 10 mark question. And the structure of this I have once more I have shown for you to draw it clearly with the microscopical character of the coriander. You have to explain very clearly the epicarp. This is the part of the outermost epidermal cells, polygonal cells, and occasionally stomata and calcium oxalate crystals are present. Mesocarp inner and outer layer of parenchyma with sclerenchyma in between. Pinkish color. Sclerenchyma is tangential and longitudinal bands can be seen, and 2 vitae only present in the commissural surface. In the dorsal surface there is no vitae are there, only lacunae which vacuole on the dorsal surface. And endocarp, longitudinal cells forming percutary layer you can see, and endosperm you can see the inside part of the seed, celluloic or cellulosic parenchyma containing oil globules and aileron grains, that is starch grains can be seen. Powder microscopy analysis you can see various cells which I was seen in the TS, like sclerenchyma layer will be seen, dark colors, pink color, groups of fusiform fibers of sclerenchyma running way and at times crossing and each other, and when thin walled lignified cells of mesocarp can be seen. Endocarp cells can be seen fragments of percutary layers. Percutary arrangements means acute arrangements of longitudinal in all directions. Thin walled lignified cells within polygonal cells of mesocarp can be seen. Vitae, very rare because very less. In the whole fruit only 4 vitae will be there. But in the whole fruit of fennel you will have 12 vitae. Here only 4 vitae will be there because only in commissural surface you have 2 vitae will be there. So vitae, few brown fragments of vitae will be seen. And endosperm, fragments of endosperm with aileron grains and oil globules. Organoleptic characters, you can see color brown powder and odor characteristic and aromatic. Spicy taste can be seen for coriander. Active constituents you can see linalool, lanolin and borneol. Volatile oil is the main constituents. Linalool, that is coriandrol and alpha penin. Lanolin, alpha and gamma terpenin and paracymene and camphor, geraniol, boreneol, fixed oil, all of them volatile oil constituents are monoterpenes. Fixed oil is also present, and malic acid, tannins, vitamin A is also as a constituent in coriander. The use of coriander, usually food coriander powder, domestic purpose cooking curries, uses, it's a carbonative, it has many flavoring agent and anthelmintic action, expels the worms, and aromatic, diuretic, stimulant, stomachic and aphrodisiac. It has some effect in the aphrodisiac. It has more stimulant, mood stimulating in the initiation of the sexual characters for. That's why it's more used in the curries, and it has very little quantity but aphrodisiac also. Oil is used along with the purgatives to prevent the gripping. So this is what, still more we have 2 more drugs which has to be discussed in the questions, like ginger, and we have one more is there. We will discuss in the next class, lecture number 3. This is lecture number 2 is completed. Thank you. This is lengthy and many classes, many important drugs we have discussed. Listen to the class clearly and write an assignment and submit to me after your Christmas vacation. When January comes 2025, I expect the Form D students to submit your assignment in volatile oil with all the drugs and details. Thank you. The small class, the 3rd lecture, I will upload very soon within 2 or 3 days. Thank you.